I'm Dan Grinnell, and I'm a student with the University of Washington Engaged program. Here to welcome you to our event tonight. As a graduate student, I get specialized training in how to do a very specific type of research. The Engaged program gave me another type of training that graduate students rarely get. I learned how to talk about my science in a way that is engaging, enlightening, and informative. You can learn more about our innovative program at engage-science.com. And now to our first guest. Jennifer McCreight is a fifth-year PhD student in the Department of Genome Sciences. When she's not busy studying human evolution, she enjoys playing strategy games, getting lost in a good book, and cooking while pretending she's on an episode of Chopped. So please give a warm welcome to Jennifer McCreight.
Thanks for that nice introduction, Dan. So today I'm going to be talking about my research and what I think is the big question. What makes us human? Now, people have been thinking what makes us different from other organisms as long as we could just look around and see everything else. We know we look different from other creatures on this planet. And there's some traits that are very human specific, so we don't see it in other creatures. For one, we have opposable thumbs, which means you can touch your thumb to each other finger in your hand, which other creatures don't have. And it actually allows us to be able to use tools and grip things more easily. We also have bigger brains, and we walk upright, something most animals don't do. So what's causing all these traits that make us uniquely human? Well, the answer is in your DNA. And today I'm going to be talking about how we can figure out exactly how your DNA is controlling these things. Now, you've probably seen this image of DNA before, that it's this helical, twisting ladder of some sort. It looks very pretty. But what exactly does it mean? What does it do? Well, all the DNA in your body is basically just a giant recipe book for how to make you. It's all the instructions you need to build a human. And a copy of this book is available in every single little cell of your body. And all that DNA, the whole instruction book, is what we call your genome. So now when you hear that I'm from the Department of Genome Sciences, you can actually know what that means. So how do we figure this out? Do we just crack open the book and start reading and say, this gives you red hair? Well, unfortunately, it's not that easy. Instead, when we open up the genome and look, we see something like this. A code. Now, what is this code of DNA? You can probably see there are 4 different letters here, A, T, C, and G. And these are the letters in the code that build the instructions for how to make you. So where do these letters come from? I showed you this helix before, the DNA helix. And all these letters are molecules that make up the rung of the ladder. So you can see there's a G here, an A here, and so on. And if we unwind this helix and read these letters from one direction to the other, we get a sequence of DNA. So these are just basically instructions. And so what are the instructions doing? Well, they tell your body how to make a protein. And proteins are the little machines inside your cells that actually get stuff done. They perform the functions that the cell does that needs to survive. So just one example of this, let's go back to hair color, like I mentioned before. Can't just crack open the book and see you have red hair. So what's going on in someone that has brown hair like me? Well, we have this one protein that's involved with this system, and it acts as a kind of switch. So normally, this shape of the protein really matters. And that's what the DNA in your cells are telling, that make the protein this shape. And the shape of the protein defines its function. So if you have a signal, let's go back. If you normally just have this protein sitting around, your cells are going to make orange pigment. So you're going to have orange colored hair. It's saying orange, orange, orange. Well, you can have a signal come in, and it fits into this protein, and the protein's switch is flipped. And now your cells will make brown pigment instead of orange. And so for people who have brown hair like me, we have 2 copies of this gene, so it's always flipping the switch to brown. That's why I have dark hair. Now, for people who have red hair, their protein has a little different shape because their DNA is different and gives different instructions for how to build this protein. So when this signal comes in to try to switch from orange to brown, It doesn't fit, it just bounces right off. And so you only make the orange because you can't flip the switch. And that's why you get the red-headed Weasleys over here. Now, seems easy enough, but how do scientists crack the code? How do we look at this long string of letters and actually figure out what it does? Now, I'm sure you're wondering why the heck are there cookies on the screen, but I assure you there's a purpose to this and it's not just making you hungry. Let's say you like chewy chocolate chip cookies, more so than the crispy kind, which is personally what I believe. Now, you want to know what exactly makes this chewy cookie chewy. It could be less eggs, a colder oven, maybe less flour, you're not sure. So you pull up the recipe for a chewy cookie, look down the list, and you're still not sure. It looks like a cookie recipe, right? But if you have the recipe for both cookies, you can do something very simple and just compare them. And when we compare these recipes, we can see that chewy cookies use melted butter, and crispy cookies use softened. And this is a real recipe in case you want to jot it down really quickly. Now, the thing is, we can do the same exact thing with DNA. So I have this very interesting looking human specimen over here and a chimpanzee. And we basically can go ahead and crack open their instruction book and get all the instructions and look where they're different. And so instead of the amount of eggs or the kind of butter you're putting in, it would be a different letter in your DNA code. Now, why does this work? Why can we actually compare these two things and learn something from it? Well, let's go back to the cookie analogy to start. And here I'm going to show you how cookies have progressed through time, with the oldest cookie recipe at the top and time going down to the bottom until we have modern cookies at the bottom. Now, I'm not getting my PhD in cookies, so this may not be totally accurate, but bear with me. So let's say we start with a really basic sugar cookie recipe, very basic cookie. At some point in time, someone decided to add cinnamon to that cookie, and you get a snickerdoodle. But that's not the only way they could have changed this original plain cookie. They could have decided to add chocolate chips instead. And then maybe there were more modifications after that, where some people decided to soften the butter and you get crispy cookies. Some decided to melt it to get chewy. And you can even have more changes after that, like adding cocoa or changing the chocolate to white chocolate. And the thing is, even if you start with this base same cookie, you end up with this diversity of different cookies at the bottom, all because changes have been gradually added over time. And that's why you can compare the different cookie recipes, because they all started from the same basic recipe. Well, your DNA is no different. A long time ago, all primates share a common ancestor, just like cookies would have shared a common ancestor. And this common ancestor to all primates has also changed gradually over time. But instead of adding chocolate chips, it's changing the DNA. So these are what we call mutations, if you've ever heard that. All it means is that maybe at one position you normally had an A, but now it's mutated to be a T. And of course, this is a simplified version. It's not just one change makes you a macaque or a human, but millions and millions of changes. And the thing to remember here is these changes are building up over a long period of time. So just to put this in perspective, our last common ancestor with chimpanzees was 9 million years ago, and our common ancestor with all other primates, all the monkeys and lemurs included, was 70 million years ago. And when you think that the generation time of a human, how long it takes for us to reproduce and have children, is 20 years, that's a whole lot of generations of humans that have been reproducing over millions and millions of years. And this time scale is why we can gradually build up so many differences. So when we get to the bottom, we see the diversity that we see today. Now there's one problem. Our genome's huge. So this is a picture from some geneticists from across the pond, and they've actually printed out one copy of the human genome. And it turns out to be 130 volumes at 4-point font, which is basically unreadable. It's ginormous, 3 billion letters. Now, so you can't just say, all right, look at the differences, because in the beginning, where do you start? There's a whole lot of genome to look through. But scientists have a little trick, and that is not all of the genome actually codes for a protein. Not all of that DNA is an instruction for how to make a protein. Now, Does anyone in the audience who is not in my department and also getting their PhD in genetics, do you know what % of the genome codes for proteins? Any guesses? Shout out a number.
1%.
1%? Any other ideas?
5%.
4 or 5? You studied coming here, right? It's actually 2%. So it is a very tiny fraction of the genome that's actually saying, make a protein this way. So what scientists did, we already knew this, we decided to just focus to start on what actually codes for proteins, because the proteins are doing the functions in the cell, and if you change that, you're going to change something about the organism, right? Seems like the obvious place to look. Well, scientists did find some things, and this is probably one of my favorite examples, so I wanted to share it all with you. Has anyone ever heard of the language gene, FOXP2, before? Yeah, some of you. Great. Well, this is the gene, amongst other genes, that scientists think are really important in producing human language, that ability that we have that so many other animals don't. And we've suspected this gene was involved in this process for a long time because we find people who have faulty versions of this gene, broken versions of the gene, and they have a really difficult time producing normal language. They have medical conditions where they can't talk as much. So we also want to look at this gene and see, how is this different from other primates? Is there anything going on in humans that makes it different? And when we compare this gene to humans and other primates, we find that there are 2 differences in this gene that humans have, and all humans have these differences, that all other primates don't. And so this is a little hint that there is something going on in this gene, probably supported by other genes as well, that is giving us the ability to have language. Though there's one interesting thing about this gene. What does it look like in Neanderthals? So just a little background. Neanderthals are another species of human. We're Homo sapiens, they're Homo neanderthalis. We lived around the same time as each other. We even interbred with them. Everyone here who is not African descent, 2% of your genome is actually from Neanderthals. And they went extinct about 50,000 years ago. Questionably around the same time humans met them for the first time, so the humans might have had something to do with them going extinct. We're not sure about that. But we have the genome for these Neanderthals now that we got from old bones. And we actually can go and look, and when we look at their FOXP2 gene, we see that it looks like ours. And that gives us a hint that maybe Neanderthals had some sort of language that puts them closer to us than other primates. That's never that easy though, right? When scientists started looking at these protein coding genes, they found that humans and chimpanzees are 98.9% similar, super similar. And it made scientists wonder, like, we clearly look so different. How can such a little amount be changed? Where are all these differences coming from if we're so similar? So what about the rest of the genome? We said only 2% codes for proteins. Now, a lot of this you might have heard is called junk DNA, where scientists either think it's doing nothing or we really just don't know what it's doing yet. And a lot of it is what we call neutral, where it really is doing nothing. But a lot of it's also super important. A lot of it is involved in what we call gene regulation. And what I mean by that is how genes are turned on or off. So it's not just the actual type of protein you're making, it's how you're making it and when. And a lot of people seem to think that genes are only turned on and off. You're either making a bunch or you're not making it all, like a light switch. But for a lot of genes, it's more like a dimmer switch where you can fine-tune the exact amount of protein that's being made. But really for most of the genome, it's more like a Rube Goldberg machine, where all these different networks are interconnecting. One gene can turn on another, one gene can turn off another, all of them in different measures and amounts. And all this complexity is really what enables your genome to build you. So what are some things that gene regulation controls? One is when the gene is turned on. Is it turned on when you're developing in your mother's womb, during puberty, all the time? Is it turned on night and day? Or is it turned on only after you eat? All these things are regulated by your genome. Where is the gene turned on? And here I mean where in the cell. a protein could be really different if it stays in the cell and has some function there instead of getting squished out of the cell and doing something outside. Where is it turned on in your body? A gene that's turned on in your brain tissue may act a lot different than one that's turned in your heart tissue or your stomach tissue, because it's all these different environments. And finally, how much of the protein is made? You can imagine if there's a protein that has some function, let's say converting one sort of chemical to another, if you have a whole lot of that protein, it's going to convert it faster. So the amount really matters. So I'm going to give you some examples of changes that scientists have found between humans and other primates that are more on the regulation side and not so much about the protein. And the first one I'd like to talk about is this MYH16, which has one change in human, just one, and it has a pretty drastic change. It has to do with the muscles in your skull. So MYH16 controls the growth of the muscles that help you chew. And if you put your fingers right here in your temple and move your jaw up and down, you'll actually feel those muscles. Those are the muscles controlled by this gene. And now the cool thing about this is in all other primates, they have a working functional copy of this gene. But ours is totally broken. This one change breaks the whole thing, so no protein's made ever. So that's a pretty big difference, not just tweaking them out, just none ever. And the thing is, these jaw muscles in other primates, they need something to attach to. Because for these other primates, their jaws are so big and they have to chew material that takes so much energy and all these leafy greens that their muscles are huge. And that's why their skulls are shaped like this, to have this little ridge at the top and ridges on the side, because that's where these muscles actually attach to the skull. And they need like a good scaffold to hold on to or the whole thing's gonna fall apart. But humans, we have these muscles, but they're tiny now. They don't really like do anything that much. And when you look at the human skull, we don't have these ridges anymore for the muscles to attach to. And the interesting thing about this is we can actually look at this DNA and figure out when this change came to be. And when we look at the time of this change, we see that it happens right around when the human brain got very large in human evolution. And so evolutionary biologists think that we're not sure the order it happened, but perhaps we had this mutation that no longer has these huge muscles, and we no longer need this kind of weird shaped skull to attach them to, and that allowed the human brain to be able to expand into a bigger shape in order to fit our brain. Pretty cool. Another example I like to give is Hackness1. Now this is a little different from the previous ones I talked about. It's not just one change. It's actually 16 changes. So all of us in the room, we have these 16 differences. We're all the same. But if you compared us to other primates, they wouldn't have it. They'd be different in 16 spots. And so again, this is outside of the actual thing that codes for a protein, so what is it doing? Well, to find this out, scientists have a little trick where we can actually take the human version of this DNA and put it into a mouse. And this is super useful because mice have really short generation times. Like I said before, humans, it takes us about 20 years before we grow up and we're able to reproduce, where a mouse can do it in months or a year. And so we can study these things a lot faster. And when we put this in mouse, we add a little tag to it. And what this tag does is it's going to show up blue wherever the gene turns on. And then they actually look at the embryo of developing mice and see, all right, where is this gene turned on? What's going on here? What makes it so special? And it shows up in a very interesting place. This is actually a mouse paw, and this is the mouse's thumb right here. So this human version of the gene is only turned on in the developing thumb of the mouse. And when they put in the copy of this DNA from chimpanzees or other primates, they don't see this. They don't see it turning on the thumb. It's only the human version. And so this makes the researchers from this project think that this little sequence of DNA is tied to the evolution of the opposable thumb, because only in humans do you see it turned on there. Now, those are some cool examples. And I know the whole talk is framed on what makes us human, what makes us special. But I have one parting little note to give all of you. What actually got me into this field is that this works for all organisms. So this is the tree of life, if you haven't seen it before. And you are here as the Homo sapiens. And this is all the other life on Earth. And the thing is, just like primates have a common ancestor, or that sugar cookie has a common ancestor, we all have a common ancestor. Whether you're a bacteria, a fungus, a plant, a mammal, a bird, we can all trace ourselves back to a common ancestor from billions of years ago. And because of this, we can use this same method to compare any organism on the planet to see what makes them special. And I'm a strong believer that humans aren't just the special ones. We can't echolocate like bats. We can't spin silk like spiders. So if any of you can, I want to know, because that'd be really cool. And because of that, using this tool, we can investigate all life on Earth, not just what makes humans special. And with that, I want to thank the Engage program for putting together this wonderful science communication class that helps grad students like us describe our research to the general public, and the college environment for supporting them, as well as Town Hall. And I want to thank Genome Sciences for being a great place to do research, especially Swanson Lab, which is my lab, and all my sources of funding for my research. And in case you're interested more about this, I do have a blog at thegenome.com where I talk about a lot of different things, including science. And I have a post that just went up 5 minutes ago that is going to have some of my favorite science posts that I've done in the past, in case you want to check it out. And with that, I would love to answer any of your questions you might have about genetics. Thank you.
I've read that different organisms, different life forms have different length DNA. And I wonder, would it be a fair statement to say that if we went back 70 million years to our common ancestor, that we would be able to see how the length of that has progressed over time? But that doesn't necessarily correspond with the fact that I've read that some animals have short and some are very long.
Yeah,
Could you explain a little bit of that?
Sure, that's a great question. It's actually very interesting because this is a big, what we call paradox in biology, in that we actually don't really see a correlation with genome size and what we would at least consider complexity. There's some simple trees and nematodes that have bigger genomes than humans do, way bigger. And it goes back to what I was talking about earlier about that junk DNA, the rest of the genome. Because a lot of it is neutral in the sense that it doesn't really do anything, but it doesn't really hurt you, it doesn't really matter if you get more of it. There isn't an incentive for evolution to kind of cut it out and chuck it out, because it's not doing anything. So you can have lots of different genome sizes that don't really mean anything biologically. But if we do go farther back in time, like to the beginning of a single cell, you would see smaller genomes there. Like when we look at bacteria, they have one little circular piece of DNA in their genome, and it's smaller than a mammal, where humans, we have 23 sets of chromosomes and much longer than a bacteria. So if you go farther enough back, it will be smaller.
How sure are we that the junk DNA is really junk?
Great question. Not very. It's always hard to say how sure we are about what we don't know. Some of it's definitely junk. Some of it's definitely neutral. And again, that's because in evolution, if it's not doing anything bad, there's no incentive for it to get weeded out. So some of it's just going to gather up. But genetics is a field that's changing like crazy, and more often we find things that it's actually doing stuff in the DNA that we didn't think it was before. A lot of projects are working to figure out a lot of these sequences that are involved with regulation and may not be as obvious. But I don't know, it's going to be an exciting field in the future to see just what, I think a big chunk will just still be junk, not doing anything, but there could be some more to figure out there.
And so how similar are we to each other?
Great question. We're about 99.9% similar to each other, very similar. And the thing there is, even if you look within a population, so we're looking at all African people, let's say, there's more difference between any two African individuals than all African individuals as a whole compared to all European individuals as a whole. So there's a lot of diversity in these groups of people.
And then that issue of like 70 million years ago, I think it was, there was a common ancestor of chimpanzees. What defines sort of like the split?
Good question. So there's never an exact moment where something becomes a new species, because when this is happening, it's in terms of populations. So you might have a huge population of primates, and a certain part of the population is generating some change gradually over time to the point where maybe you have two subpopulations that are slowly becoming different. But when you look at that right when it's first happening, they're still pretty similar. And so it's kind of like, at what time point do you look and decide, oh, they're different enough? For biology, when we define a species, we say, can they breed or not? Which is kind of like an arbitrary cutoff, just so we can label things. But there are problems that come with that. Like, we used to call Neanderthals different species, but we found we did interbreed with them. So are they truly different? So I guess the answer to that is there's never one clear cutoff point, because it's a process that's always occurring in time.
Thank you.
This question is kind of related to the 98.9 and the 99% likeness. What puzzles me, and I'm not understanding something here, is that we share 2% of the genome with Neanderthal. So why are we 98% different when Neanderthals are so much closer to us than chimpanzees? Is that clear?
Yes, I see where I misled you. So when I say the 2%, that means if you're looking at a human genome, like let's say you're looking at my genome, throw my genome up on the screen. Of all the DNA in my body, 2% of it came from a Neanderthal originally. But the thing is, Neanderthals are really similar to us because they were the closest related species through evolution, closer than chimpanzees. So if you looked at a Neanderthal genome on its own with no interbreeding, they'd already be like 99% similar to us. They'd be more similar than chimpanzees. And so when you're cutting out that 2% of their genome and putting it in us, it's already so similar that it's not going to make like a huge difference. So did that answer your question? Yeah, it's not that we share only 2% of our genome with Neanderthals. It's that of our modern genome, 2% came originally from some Neanderthal, but because that 2% is related through evolution, it's still going to look very similar to us. Does that answer your question? Okay, great.
Yeah, so I don't know if this is in your field, but I'm curious about the on and off switch.
Mm-hmm.
And I've read, like, for example, genetic diseases, and sometimes we're like, oh, you're destined to have this disease from your genes, but they're kind of changing and saying, well, you actually have some influence, and you can actually, with your consciousness or your thoughts and so on, you can go in and turn it on and off, those switches. So all depending on how, if you do that, you produce certain genes and certain diseases, and that it has actually nothing to do with your genes. It's you that has an influence of what's going to come out of your genes, mm-hmm. what proteins are produced.
I would say vast majority of the time, well, okay, there's two parts to this. Some traits are more controlled by genetics than others. So, for example, there's this disease called AMD, where it's a type of blindness where you go blind later in life. And if you have the copy of those genes that are bad, you're pretty assured you're going to get it. Like, it's a pretty high chance. But there are other traits that are influenced by genes and their environment, like height, where there's a genetic component, but if you eat really well and you're in a good environment, you're going to grow better than someone who's malnourished and all that sort of stuff. But when it comes to like consciousness or willing yourself to like turn genes on or off, that's not really something that will control what genes are doing. But maybe if they're thinking of it as like you can change certain things about your life that might change your biology. So like, if you eat really well versus eating terrible all the time, that might be sending signals to pathways in your body to do different things. But it's all biological. You can't just like will yourself to turn a gene on or off, but you can maybe change the inputs that you're putting into the system. Does that make sense? Yeah.
Thank you.
Well, thank you for a very enjoyable lecture. Could you talk some more about the relationship between proteins and the genome, how scientists identify associated genome with a protein, and how many proteins have been identified, and have all of those been mapped on the genome at this point, or is that still ongoing?
Great question. Humans have about 25,000 different types of proteins that the whole genome codes for. And I would say for humans, something that's really well studied, and like mice, we know most of those proteins. There might still be some that are a little harder to figure out than others. And the way we figure that out, proteins, well, so DNA is a code, right? And there are rules in this code for how to make a protein. And we've learned what those rules are. For example, proteins will have specific sequences, and we know what those sequences are now, that always occur before the protein. And it's a way for the gene to say, hey, start here. Like this is where this gene starts. And so we can look for those sequences, and that gives us a hint where proteins may be. Did that answer your question?
It does.
Okay, it helps. Okay. You can always come talk to me more afterward too, talk more in depth. But for things other than humans, yeah, we don't have all the proteins figured out all the time. It's still something that basic research is looking into.
It seems like you invited questions beyond human genetics.
I'll try.
So, if that's true, could you comment on the ecosystem concept of the genes of spawned out salmon being in trees?
I know nothing about that. I'm sorry. What is it? It's that.
Well, it's the notion that the trees in the river ecosystem where salmon spawn and die, and their remains are distributed throughout the ecosystem, including the trees in the area. And I've heard that in a number of places. A recent documentary, I was kind of curious if somebody who is in the field could comment on that, to what degree that's literally true. And
My gut would be that I don't think the tree should be able to absorb any DNA from.
That's why I was asking.
Okay, yeah.
And two or three different seemingly reliable places.
Yeah, so think of it this way. When we take like an apple, you're gonna eat an apple. That apple it's made of cells, and each cell has DNA in it, right? Well, when you eat that apple and it goes in your body and goes in your gut, your stomach acid totally annihilates everything. It breaks everything down. So whenever you hear stories about, oh, you're gonna eat some weird DNA and it's gonna get incorporated in your body, it's not true. It's just because it's gonna get digested along with all the other DNA you eat. So with the salmon thing, I think maybe they mean the nutrients from the salmon's feeding the trees, because that would be true. If a salmon died by a tree and broke down, the tree could use it to grow, but it wouldn't be absorbing its genes or anything like that.
A question about what genomic science might do in the future. The health of the genome, of the human genome, it seems to me, when we evolve to the point where healthcare can overcome many genetic deficiencies, those deficiencies get passed along, and that builds up over time. Can genomic science at some point look at the state of our genome and maybe make some corrections to it?
It's a great question, and the answer is someone actually just did that. I don't know if you've been following, but there's some research groups in China that they just for the first time genetically engineered a human embryo where they inserted a good sequence for a gene to replace a broken sequence that would have caused disease. And now these embryos weren't raised into humans or anything, it was just in the lab. But it's possible to do that with the technology we have nowadays. If you want to read more about it, it's called the CRISPR system, and it's CRISPR. Very interesting. But yeah, it's definitely possible. So There are clearly risks associated with that. I know in the study, a lot of the stuff they were trying to insert was inserted in the wrong spot of the genome, which is never good because your genome has a really strict set of instructions and you don't want to be inserting things in the wrong spot because sometimes that causes things like cancer. So we really want to make sure we have it right before we go too far. But it is within the realm of possibility, for sure.
You took us back 70 million years to a common ancestor. If you took that back, excuse me, several billion years, does that imply that life arose only once?
Ooh, good question. Not sure. As the farther you go back, the more it's going to get fuzzy, right? So it's easy to compare all mammals or even all mammals, birds, reptiles, anything that's what we call eukaryote. It's easy to compare those, and clearly we're related. So if you take all plants and all animals, you can make trees like this, and you see the relatedness. Like we share 50% of our genome with bananas because that's just the 50% that it says make a cell. You know, all life is going to share some basic stuff, just like that cookie recipe, where there are going to be some things that all cookie recipes have. But yeah, I wish I could go in a time machine and go back and look, because there's a chance it evolved more than once, but there's also a chance it evolved many times and then a lot of those died off and are not still around today. But at least from when we look at life today, it seems to have some pretty basic similarities that make it seem like maybe one.
Thanks.
Yeah.
So the DNA, is there any chance that it is going to change? I heard something like from 10 to 12 strands, or I'm not sure about the details, but what about that? Are they talking about the human DNA actually changing, or the possibility of that?
DNA is always changing in the sense of the actual sequence, so those A, T, C's, and G's. You're constantly getting mutations. Everyone here is a mutant, I'm sorry. And those kind of like sequence changes happen. You can have some bigger structural changes. Like there's this thing called copy number variant, which basically means instead of just having one copy of a gene, you can maybe have 2 or 3 or 4 or 6 or whatever. And there can actually be variation where I might only have 2 copies, you might have 6 copies, you might have 8. But you wouldn't see any really big changes like DNA is probably always going to be a helix with those kind of rungs of the ladder. You probably won't have 3 or 4 strands instead of the 2. That's pretty, what we say, conserved. Like it hasn't changed since even you can look at bacteria and it looks the same way. Does that kind of, yeah, thanks. Any other questions? Well, thanks so much for having me. Thank you very much.